right, welcome everyone and welcome to this program by Studium Generale of the Eindhoven University of Technology. Live here from the Blauwe Zaal, but we are also working on a new live stream setup, so also a very welcome to everyone who is watching the live stream. And if you are wondering how to register for SGNUs, later on in this program we will make an online form available in the chat. So keep an eye on the chat, and of course you have to watch the whole program to get your credits. So let's go to the topic of today, fermentation. And I was wondering, who of you, the people here in the room, is doing fermentation themselves? Who of you is experimenting with fermentation, with fermentating foods? I see one hand going up. 2, 3, 4. Okay. Okay. About 10 people here in the room. Maybe there are also people watching online who are doing fermentation, food fermentation, or other things with fermentation. You can let us know in the chat. Fermenting food is it's very popular nowadays, and fermenting food, it's healthy, it's tasty, and this old craft is on the rise again. But the application possibilities of fermentation hold even greater promises. And that is why we have invited fermentation specialist Professor Jeroen Hugenholtz for today to discuss how bacteria, yeast and fungi can play a crucial role in the solution of major world problems. And then you have to think of world problems like solving the world food problem and other problems that have a major impact on the environment. And then think of the production of palm oil and plastics and cosmetics. So, a very fascinating topic to discuss today, and I'm very excited to hear more about this. After the lecture, there's time for questions, and also for the people watching online. If you have questions, you can ask your questions in the chat. But for now, please your attention and give him a warm welcome, Professor Jeroen Hugenholtz.
Okay, thank you, Gijs, for this nice introduction, and the audience for this applause. It's a pleasure for me to be here. I don't come here very often, and I think we should do much more together with the different institutions that I represent, and together with the TU Eindhoven. What I will do in the next 25 minutes or so is share with you some of my thoughts and some of my experience, and also some of the running projects on the area of fermentation, and especially to expand the world of fermentation. What we should realize before we start, and also it's also part of the things that we do, is that fermentation is all around us every day. So if you look at the environment, for instance, we have these cows and other rumen bacteria where fermentation is going on all the time. And this is nowadays, there's a lot of attention to it because of this methane and this gas emission, especially in the mouth, but also from the back. And this is building up in the environment and causing major environmental problems. But here you see also the similar fermentations are taking place daily in swamp areas all around the world, and especially in places in the world where the temperature is going up. This is actually increasing the amount of methane emission. This is also presenting a major environmental problem at the moment, all as a result of this fermentation process leading to methane production. We see it also in industry. Fermentation is being used, and we see here an example of a digester of a municipal solid waste processing plant. This is in Wijster, in Drenthe. We see here also an example of a huge factory where bioethanol is being produced. Ethanol, also a result of a fermentation process. And here this is a factory in Brazil where sugar cane is fermented to bioethanol as a fuel. And then, of course, fermentation is used and consumed by us daily. And we have the products that you probably all recognize here. You have the alcoholic beverages. You have the bread, all where fermentation is taking place with yeast, resulting in alcohol formation and also resulting in gas formation, leading to the rise of the dough, which is an essential process in bread making. But you also see here products like sausage and cheese and yogurt and buttermilk that is a result of lactic acid fermentation. So these are lactic acid bacteria that ferment the milk into organic acid, in this case lactic acid, leading to a product with longer shelf life, but also with improved taste. And then let's say the organisms that we're dealing with, this is just an example here. This is yeast. And so when you look under electron microscope, you really zoom into these organisms. They look like this. This is the yeast called Saccharomyces cerevisiae. So that's the brewer's yeast, which is also used for bakery. And this is basically, if you are into biochemistry, this is the fermentation path that we are dealing with, where sugar and glucose or other sugars are being converted into ethanol and CO2. So the CO2 is for the rising of the dough, and the ethanol is, of course, the important product in alcoholic beverages. So if you look at fermentation, well, the two major ones that we know in the food industry and now also in the industrial industry, I have already described those, are the alcoholic beverages. So that's a yeast mainly that is doing this, and there are this is the most well-known yeast, but there are various other yeasts that are also used in this application area. And then we have the lactic acid fermentation, and we have organisms like lactobacillus and lactococcus that are used for either cheese or production, or for production of things like sausages or sauerkraut, olives. All those products are a result of a lactic acid fermentation. But those are not the only ones. I mean, there are many other types of fermentations, some of them that we actually don't even know, but there are several ones that are already quite well described because they are being used for different purposes. So we have the acetic acid fermentation, where organisms like Acetobacter and Gluconobacter play an essential role, leading to formation of acetic acid. Citric acid fermentation, that's a fungus called Aspergillus, which is the main organism being used for that. And of course, you know that citric acid is used as an additive also in many food and beverages. We have propionic acid fermentation, which is again another type of fermentation where an organism called Propionibacterium is involved. And this is well known as the typical nutty flavor in Swiss cheese. And the Emmentaler, Gruyere, Maasdammer cheese, that's where this organism is playing a crucial role for this typical flavor that is being formed there. And then we have various other fermentations like succinic acid fermentation. I will come to that in a few minutes. We have a fermentation called ABE, acetone, butanol, ethanol. This is an organism producing a mixture of these compounds. And that is an organism called Clostridium acetobutylicum. I also come to that organism later in my talk. And we have, of course, which I already described in my pictures, the fermentation leading to methane production. And those are strictly anaerobic organisms that can convert all kind of carbohydrates and smaller organic acids into methane as their main end product. But we also have organisms that actually can consume these methane. So they actually oxidize the methane to CO2 and an organism called Methylococcus. We have also organisms that are known to convert syngas, which is also a major exhaust from the chemical industry. And there are organisms that are known to convert that syngas into other compounds like acetic acid and ethanol. So we are dealing with a whole set of fermentations that can be applied in several ways. So the way that we are inspired in the work that I do is about finding new ways to use fermentation for our benefit. And we are basically inspired by something like this. This is what we call the periodic table of fermentation. And here all the fermentations basically are summarized that we know basically from the food industry. And that's traditional industry that has been out there for ages. And many of those have been described in some detail. So we know what organisms are involved there, and we also know what kind of fermentation is taking place. And those are the organisms, the source of the organisms that we use nowadays for all kind of new processes that we are developing. And then we are talking about producing all kind of consumer products that we use daily, and not only food, but also all kind of chemicals, plastics, biofuels, etc., where we try to replace the fossil fuel as a source for these chemicals with a fermentation process. And then we are inspired by all these organisms that have been described in these traditional products. So if you then look at this schematically, and this is a sort of a simple way of depicting this, is that we take an organism, and in this case the organism is this yellow oval in the center. This organism is able to convert sugar, or maybe a whole biomass waste stream from the agri-food industry, or even a gas, as I described just a few seconds ago, and convert that in a fermentation process to an organic acid, for instance, lactic acid, or an alcohol, or butanol, or something similar. But they do more than that. And these organisms basically also produce vitamins in some cases, or they produce, at least all of them, when they grow, they produce their own protein. I will go over some examples where we can use these specific reactions leading to these side products for our own benefit. And these are sort of in the category of foods. And here we are also looking at some of the non-food applications where fermentation can be applied. And well, one of the ones that I'm not going to discuss in more detail, but the protein is, let's say, one of the upcoming areas where basically we are looking also in the feed industry, where protein is now being supplied by soy coming from faraway countries. And we know already from literature and from experience that the amount of protein that is produced by all these industrial fermentation process, and in the future that will be even more, there's enough protein there to feed all the livestock that we have all over the world. So that means that this is a potential very interesting source of protein for the food and feed industry. Okay, so I will go over just briefly over these 5 examples about preservation, about using fermentation to produce low-calorie sweeteners, oils and fatty acids produced by fermentation process, and then also some non-food examples like bioplastics and solvents and biofuels. And I will take just a few minutes for every example. So we start with preservation. If you look at the food industry, let's say the traditional way that fermentation was developed and that we learned more about this fermentation, is the preservation, because the production of either alcohol by an alcoholic fermentation or an organic acid like lactic acid or acetic acid actually leads to the accumulation of these compounds and leads to the inhibition of growth of all kind of undesirable spoilage microorganisms. Fermentation is actually an original preservation strategy, and that is how perishable foods, fresh foods, could be used much longer by humans that were dependent on these products. But nowadays we all have refrigerators, etc., so we are not anymore dependent on fermentation for preservation. But we are still looking in some cases for a longer shelf life. So one of the things that we're working on is to increase shelf life also of some of the products that we have in our refrigerator. And for instance, you probably know when you buy fresh fruit like strawberries, that if you keep them, especially at a summer day for a few days, you will have a nice white layer of mold on top of them, and there's not much you can do about that. Well, that is something that basically in this industry of food is sort of unacceptable, so we are trying to find solutions there. And we see those solutions also in using so-called protective cultures. So these are lactic acid bacteria. They produce lactic acid, but they do more than that. They produce also some additional components that have been analyzed in some cases, and we see some specific peptides that are being formed. We see some phenolic compounds like phenylactic acid being formed, and sometimes some antifungal proteins or compounds like cinnamic acid that are being formed and that have demonstrated antifungal activity. And this is something that you can use for your advantage, for longer shelf life of the products, but also for so you can keep these products longer in your refrigerator. And here are examples of how this works. And you here see different fruit juices where you see, in this case, white, in this case, a little bit blackish fungi growing on the surface of this fruit juice. But here's an example, and here also, where it's basically free of these fungi. And here that we added a little bit of this protective culture. So this was producing something that inhibited the growth of these fungi and leading to a much longer shelf life. Well, this same kind of approach can work on, let's say, fresh strawberries. So we sprayed this culture on top of these strawberries, and then you see in some cases that there is quite a good protection, while here these are completely covered by fungi. So that is, let's say, the way that this research is then advancing in this area. And based on these kind of studies, you select cultures, you select procedures that leads to protection against fungal spoilage. This can also be applied, for instance, on fresh flowers. I don't know if you have ever noticed that, but if you take roses that have not been treated with certain antifungal agents, you get that they start forming a whole layer of fungi around it, which does not look very well in your living room. But if you use and spray them or submerge them in a protective ferment, as we call it, then you can get much longer shelf life of these flowers. So also for this kind of category of products, there is a very interesting possibilities. And one of the things that we are exploring now is to also use this kind of approaches for protecting crops on the land. You might have read and heard about developments in South America and also in Africa, where banana plantations are being attacked by fungal spoilage, and there are all kind of restrictions about agents that can be used nowadays. And so these are more or less natural agents that we think and we hope can be used for protection against these kind of diseases. Okay, the next topic, also related to food, and this deals with, let's say, our societal problem of obesity, about consumption of too much calories. And one of the nice things about fermentation is that fermentation, many of the organisms that we use for fermentation, they actually consume sugars. Now, if you do that for products and you completely convert the sugars, then of course the product is much lower in calories, so that's good. But unfortunately, it doesn't taste very well because you need some sweetness for your liking. So what we do with a fermentation approach is that we convert some of these sugars, like sucrose, glucose, fructose, into low-calorie sweeteners. And there are several examples of naturally occurring low-calorie sweeteners, like the sugar alcohols, mannitol, sorbitol, erythritol, and also some di- and trisaccharides like trialose or tagatose, or isomers of sugars that are not basically metabolized by the human body, but still are sweet, and then in that way can actually contribute to lower calories. And then you can basically look at a scheme like this, because we know that fructose is the most sweet sugar. Sucrose and glucose are less sweet, but you can replace that with some of these low-calorie carbohydrates, offering similar sweetness but much lower calories. And you see, for instance, if you take a milk product which contains lactose, which is not very sweet, actually, lactose, and you convert that into something like erythritol, you actually can get an increase in sweetness. So that is also a concept that can be used for the benefit of the consumer. So then we are, of course, have been looking for organisms that can actually produce these kind of low-calorie sugars. And there are several yeasts and also bacteria that are known to produce these. And we have actually been able to use some of these fermentation processes to produce products which are actually much lower in calorie and still taste like the original product. And this is, we have focused quite a lot on the yeast called Moniella, which is a natural resident of honey. So it's used to live and flourish in environments with very high sugar. And basically this high sugar is converted by this organism into erythritol. Well, and then the concept that we have been working on, and where there's lots of attention in the food industry, is to convert basically different fruit juices where there's 10% or more of sugar, and we use a fermentation process leading to very little flavor changes, but the sugar is completely converted partly into erythritol and also in some other compounds, including the yeast themselves. And this still leads to a product where that still tastes like the original fruit juice, but the calories have been tremendously reduced. But this is the type of area, and let's say a new area, where fermentation can play a major role. Well, we can also look at other ways of increasing or decreasing calories and maintaining sweetness by looking at compounds that are produced not directly by sugar fermentation, by conversion of some proteins where different peptides are being formed. And using human taste receptor lines, we can actually analyze for the presence of these peptides that have sweetness, and then you can actually, by production of these peptides as a result of hydrolysis of protein, you can also increase sweetness of a fermented product. Okay, the next topic that I want to cover is the use of fermentation to produce oils or fatty acids. And here we use some yeasts that we know that actually accumulate huge amounts of oils. So if you look at these under the microscope, you see these oil dripping almost out of the cell, and on the agar plate you see these colonies which represent a whole group of these yeast that looks very oily on the surface, and that's because the whole biomass is composed of more than 50% of oil. Well, this basically the oil, if you refine it, so if you basically extract it from the microorganism, it looks very much like sunflower oil or olive oil or palm oil. So the type of organisms that we are looking at are yeast called Cryptococcus. It's actually the highest accumulator of these oils. Or there's also a food grade variety called, which then is allowed to be used in food because of a history of safe use, called Yarrowia. We also know various microalgae. And the nice thing about these is that they produce much higher amounts of oil than all the vegetable and plants that we know that accumulate oil. So that means that you have a much more efficient process. And we have the ability to make and to control the amount and also the type of oils that are being made. So we can basically change the chain length of the fatty acids built in these oils or the saturation level of the fatty acids. So these oils that are produced, here we see the typical composition of these yeast, and it is sort of in between palm and sunflower oil with respect to composition. But we also know that from some of the studies that we did that you can actually look at the individual contribution of these different fatty acids, and you can change that as a result of fermentation conditions. And so what we are doing now, and there are actually companies that are starting up, and I'm actually involved in one of those companies, where we are basically setting up an alternative source of vegetable oils, now based on a fermentation process and not based on palm or coca or coconut or sunflower. And that means that we are actually providing a much more sustainable solution for the production of these oils, which are massively used all over the place in our society. And the application of these oils is, of course, in products like mayonnaise, but also in dairy products and in chocolate. And it's used for baking and frying, but also for dressings and sauces and creams. But also outside the food industry, it's used as a surfactant and emulsifier. Fatty alcohols, a reduction product of these fatty acids, is used also in a lot of cosmetic products. And then we know the PFAS, the omega fatty acids that are used as a nutritional component, for instance, in baby foods. So, as I told you, when you are growing these yeast under different conditions, for instance, if you change the fermentation temperature, you get a major change in composition of your fatty acids. You get much more saturated, unsaturated fatty acids when you look at low temperatures of cultivation, and you get much longer fatty acids when you look at higher temperatures. So we have actually a way to control that, and that's not the case when you use palm oil or when you use sunflower oil. So this is a very interesting alternative, and there's major attention now to switch to these kind of oils produced by fermentation. Well, you can also use this as a platform, and then you have to go more in the direction of genetic engineering, where you can look at introduction of new pathways and also changing the pathways of oil production, and then you can make, let's say, the more unique fatty acids also, and then this platform can be a source for that as well. Okay, the next topic I would like to cover quickly is the production of bioplastics. And also here, microorganisms can help us. We know of certain microorganisms like Pseudomonas is known to produce a compound which has very much polyester-like activity. It's called polyhydroxyalkanoates. It's a storage molecule, which this organism produces in times of need. So when it is, well, actually not in times of need, but it can use that when the growth substrate is deficient, and then it can actually hydrolyze this storage molecule, and then it can grow on that so that it will survive. But this PHA has all kind of plastic properties. So this is now being considered as an alternative to some of the plastics, fossil fuel-based plastics nowadays. These organisms look under the microscope like this, so we have all these plastic bodies included in the cell, and you can easily isolate them, and then you have a nice biological plastic source. Well, we can actually also influence the properties of these plastics by building in longer fatty acids in the polymer, and then we get much more flexible molecules that can also be used as wrapping and packaging material. Well, here are some examples of products that you can make. You can use it in a latex, you can make a rubber, you can use it for harder plastics like cups, etc. Now, this is not the only bioplastic where fermentation can play a role. Lots of you might have heard of polylactic acid. So lactic acid, of course, direct product from fermentation, but also like polyglycolic acid. Succinic acid is used also in a polymer, and different organisms have been found and are being explored for the possibility to produce this on a large scale. Okay, the final topic I wanted to cover before I finish is the production of biofuels and solvents and some bulk chemicals using also a fermentation process. And here, one of the organisms that we have been working on for many years in Wageningen is the Clostridium, either the one called Beyerinckii or the other one is called Acetobutylicum. But the very special feature of this organism is that it produces various solvents like ethanol, butanol, and acetone in large amounts. And it can grow on all kinds of carbohydrates. And it does this anaerobically. And we have seen and we have demonstrated that you can actually grow these organisms on all kinds of waste streams that are abundantly available in the world, like seaweeds and sugar beet pulp and wheat straw pulp and various other substrates. And we have basically developed specialized fermentation processes that can completely convert these waste streams into these solvents, alcohol, ethanol, butanol, and acetone. And butanol is a very good fuel. And we have seen examples where we have actually driven cars based on butanol that has been produced by this fermentation of seaweed. Well, the same platform or similar platform is now also being explored for conversion of another type of waste, which is gas. The waste gas coming from different pyrolysis, so heat destruction of biomass, and also in the cement industry and the electricity industry, lots of syngas is being formed. And syngas is actually a combination of CO2 and carbon monoxide and also hydrogen. And this syngas is, of course, a major contributor to green gas emission, but it is also toxic. And there are organisms actually that can convert this syngas into beneficial compounds. And this is something that we are exploring in a large consortium within the Netherlands. It's called SynValue. And there, what we do is we take specific organisms that are able to convert this syngas into compounds like acetate or ethanol, shown here, and then combined with other organisms that can, again, use this acetate and ethanol for production of, for instance, propanol or hexanol or even longer alcohols, we can actually develop a whole microbial shelf of combinations of organisms that can produce this whole list of chemicals that are very important in the chemical industry. And this is something that we are working on, and this is what industry is asking for a more sustainable solution for the production of all these chemicals that are currently made from fossil fuels. So in summary, well, my story started with that fermentation examples are everywhere, and we are actually exploring these fermentations in all kind of different areas, like food protection, crop protection. And nowadays you hear a lot of precision fermentation. That's the term that is being used, where you take a selected organism and use it for a specific purpose. So fermentation can be used for calorie reduction. I described that in a little bit more detail. We can use it for production of natural oils as a replacement of palm oil. You can also use it for bioplastics and biofuels and solvents. And with that, that's the end of my story. I would like to thank you for your attention.
Well, Professor Hugenholtz, thank you for your talk. Yeah, indeed. Yeah, there's a lot of interesting information to share, but we have not the time to go to discuss all the details. But we still have time for at least one question from the audience here in the room, or maybe the audience online. You can go to the chat and ask your question there. Is there someone in the room here who has a question for Professor Hugenholtz? Yeah, there's a question over there, and the mic is coming your way, the catchbox mic.
I talk with this? Okay.
Yeah, keep it closely to your mouth, then it works best.
This is quite funny. Okay, Professor, you talked about these microorganisms as a more or less natural way to, for example, spray the fields and treat agricultural products. Can you explain me a bit more, like how are these cultures produced? Are they actually more sustainable in the long run? Are they like really more natural so we can use them?
Well, what I can say about that is that these organisms that we are studying in this respect are all natural isolates. So there's no genetic modification involved, but we have screened them for their ability to kill fungi. Okay. And then those organisms, then the ones that is most powerful or active against the specific fungus that you want to kill, we select them, and then you can do two things. You can either apply the organism on, and spray it on top of the surface of your crop, or you can do a separate fermentation, make fermentation fluid, and then spray this fluid over the crop as a crop protective agent.
Yeah. I mean, I even use some microorganisms, for example, in my composting at home, and I wonder, like, the way they are produced, the way they are isolated, is it like really more sustainable? I mean, on the long run, like for.
And is it a sustainable solution in the end?
Yeah, like for the actually like spraying whole fields and like using it as a kind of a solution. Is it being made more sustainable?
Well, basically, if you talk about crop protection, so that's sort of the example that I implied. Nowadays, they use all kind of quite toxic chemicals. And those are sometimes remaining in the soil for a long time. And they are definitely not good for the environment. And we are here looking for replacement of natural products from a fermentation process. These are all compounds that are eventually degradable. So that means that in that case, you certainly have less impact on the environment.
Okay, yeah, that's exactly what I wanted to know. Thank you.
Okay, thank you. That was a good question. Looking at the time, unfortunately, we have to wrap up this session. If you still have questions, you can come down and just ask your question in person to Professor Hugenholtz. For everyone who is watching online, and if you would like to register for Edgy and Use, you can go to the chat. There's a registration form. You can fill it in, and then you get your credits. Thank you, everyone here in the room, for being here. Thank you, everyone watching online for watching online. Keep an eye on our website for the program that's coming up, and also for which programs you can watch online. So stay safe, and hope to see you offline or online anywhere. Bye-bye.